from the UCLA Herbarium, Patterns of Spontaneous Sidewalk Vegetation in Urban Los Angeles.
I think that one's only on Zoom.
Oh, okay. That may just be on Zoom. So botany lunch, if you're interested, look online. You should be able to find out. MVZ lunch next week is Eric Enbody, who's a postdoc at UC Santa Cruz with Russ Corbett DTIG. I heard him speak at a meeting in Asilomar in the spring, and it was one of the most impressive talks I've heard in years. It was just remarkable. He has whole genome sequence data on Darwin's finches in collaboration with Peter and Rosemary Grant over decades. And he gave just a beautiful talk on this. So this is going to be, I assume this is what he's going to talk about. After hearing that, I immediately invited him to come up from Santa Cruz. So he'll be speaking next week. I'll be sending out an email if anybody wants to meet with him one-on-one. He's a nice guy. He's a postdoc. He looks like an up-and-coming person working on Darwin's finches and genomic data. The only other announcement, yeah, Charles.
Yes, there's also a seminar, the Wildlife and Conservation Biology Seminar in Aspen by Zoom. Is a first-year grad student from Bashir's lab.
She's here.
She's here?
Yeah, in front of you, yes.
The title is Land of the Endemics, Combining Science and Compassion to Protect Threatened Yet Overlooked Wildlife in the Indonesian island of Sulawesi.
Thank you.
Wonderful, thank you.
Your name is, sorry? Okay.
Yeah, thank you, sir.
Free advertisement. All right, any other announcements? Yeah, no, it's all right. So we are going to host another night at the museum on Tuesday, November 15th. And this is not something that's open to the public or even the MVZ community, except for a few people, which I'm going to invite, but I want everyone to know about it for a couple of reasons. So this is something that we've done once before, in which we have sort of a fancy hoity-toity event and invite a very curated list of potential donors to the museum. And we have wine and cheese. We're going to clean up the museum before this event, so I'll be sending some emails around asking everybody to clean up. We'll have we'll give them tours of the collection. The faculty, I've asked Rory to give a brief talk. I'll give a brief talk, and I'll be asking 4 graduate students, like we did last time, to give a 1-minute talk each. So sort of an elevator pitch standing in the gallery. This was spectacular last time we did it. It worked very well. It raised a lot of money for our endowments. And so this is something we had hoped to do sort of every other year or maybe every other year. But then the pandemic hit. So we haven't done it. It's been 3 years since the last one, but it was very nice. We followed up after the people visited by sending everybody who came a photo album that we've made of the MVZ. And I think everybody has probably seen it. But I asked Carly to leave out a copy in the front, or a couple of copies in the front office. She sent out an email this morning asking everybody to send photos. And so we did this 3 years ago, and I think people didn't have a sense of what it was going to be used for. So I just want to be clear. The photo album that Carly produced last time was spectacular. It's like a coffee table book with pictures of organisms and people in the field. And we're going to do the same thing again, but we want all different pictures because we're going to send this to some of the same people. We don't want them to have the same book twice. But it's an opportunity to showcase the organisms that we work on and our own work in the field. So if you haven't looked at it, look at the books that are in the front office. And if you have great pictures, we want really, really nice pictures, send them, upload them to the folder that at the link that Carly sent around. So the timeline is short. We want to send this out right after the event in November, which means we need to put the book together in the week before that. So let's try to have a good showing of that again. Okay, I've taken more time than I intended. I'll turn it over to Jim, who will introduce him.
Alrighty. Well, it's my pleasure to introduce Umilaela Arifin, who's today's speaker. Just by way of background, Umi, we call her Umi. Umi, Chumi Umi.
Yeah.
Yeah. So Umi is, Charles will understand the Chumi Umi. So Umi did her undergraduate studies at the Bandung Institute of Technology, ITB. And she also did her master's there. And both as an undergraduate and as a master's student, she worked with a longtime collaborator of mine, Dr. Joko Iskandar. After she finished her master's, she went off to the University of Hamburg, or Uni Hamburg, University Stadt Hamburg, whatever it is, Uni Hamburg, to work with Alexander Haas, a famous tadpole biologist, for her PhD work. And after she finished her PhD, she had a 9-month postdoc there, also working with Alexander.
Mhm.
And then since that time, she's been a Marie Curie postdoc. It's a 3-year postdoc, which is split time between here working with me and working also with Alexander again at the Leibniz Institute for the Analysis of Biodiversity Change in Germany. Unfortunately, Umi was going to have more time here, but the COVID pandemic had kind of a severe impact. But we're lucky that we were able to get you for 9 months, 11 months. Would have been better if it was a year and a half. I've known
2
Umi
years.
since 2004. And so Umi joined one of my expeditions. We had a big expedition in 2004 to Sulawesi. And she participated on that trip. She was an undergrad at the time working with Pat Joko. And since that time, I think we've been in the field together maybe on
I don't
8
know.
expeditions.
I did not count it.
I know, I was trying to
Too
count. I
many
mean,
times.
many times. And of course, Umi has been in the field many other times besides the time she was in the field with me. I was just thinking to myself that there may not be another person who's done more herpetological field work in Indonesia during the last 18 years than Umi. She's an extremely intrepid field biologist. She's also an incredibly delightful field companion and a real problem solver in the field. And so
Thank
we
you.
love having Umi on expeditions because she solves so many problems for us that we can't otherwise solve on our own. We're really dependent on her for that. Umi works on amphibians, she works on tadpoles. Her work that she's going to be discussing today involves gastromyzophorous tadpoles. So the title is Life in the Fast Flow: Phylogenetic Systematics, Diversity and Biogeography of Asian Ranids with Gastromyzophorous Tadpoles. I guess I should mention the fun fact, right?
Sure.
The fun fact involves you're befriending a tiger in Sumatra while doing fieldwork there. And I know befriending is a yeah,
A
that's a
quotation mark.
euphemism for you were being afraid of a tiger that was visiting your tent in Sumatra. So Umi, take it away.
Thank you. Thank you so much, Ben. Thank you so much, Ben, for such a long and nice introduction. I'm thrilled. And yeah, it's also like really nice to be here, standing here. Almost at the end of my time at MPZ. I'm sad, but I've been really like have a great time so far. Yeah, I'm just couldn't explain more. So yeah, don't have to read the title. You can, but maybe it's also like really it's difficult to pronounce this because it was also like difficult for me at the beginning, but then don't worry. So yeah, Okay, so what is gastromyzophorous tadpoles? Let's start with this. So you need to know like, the tadpoles that I'm studying. So gastromyzophorous tadpoles is a very unique tadpoles that has like this really like unique adaptation because they live in such an extreme habitat, which is like fast flowing stream, the stream that you see in the title. Because like here, so this is like the abdominal sucker. It's looks like a suction cup or like a belly sucker. So it's basically no other frog like develop this kind of adaptation. Only like in this like some ranids in Asia and some American bufonids. And so the Asian one where I'm from is the one that I'm focusing on. So just to make you have a little more understanding on how they actually like live and how the suction cup like work to help them swing on the rock is you can see this in the video. Like, so there's like really fast flowing stream, and then so like the tadpole is just like hanging in there, and then just like it's really strong, and then so it just like it survived. But then maybe you're questioning whether like the sucker is like just to hanging on or it's also have some other like function. Like how do they eat? How do they move? Do you think they use like the sucker? No. No. So the sucker is literally to just like chewing on this frog, but then like for moving, they're like using like the mouth. So they're like basically like grasping the algae, possibly like, so eating and like moving, like dragging themselves, you know, like around the rocks. And so this is like very strange thing and bizarre thing. But then I think this is like what made me like finally falling in love with this tadpoles. And yeah, so then let's move forward and like, what about like how many species are there, and then like where they're distributed. So then originally all ranids with gastromyzophorous tadpoles was placed into like one genus called Amolops until 1991. Someone from China decided to split them into three based on mainly a tadpoles characters and some adult characters. And so like these three genera were like distributed mainly like in the mainland of China. It's like ranging from Nepal, Himalayan region, all the way to peninsular Malaysia here. And that is like mainly like Amolops. So this is like the mainland genus kind of. And then back then, before my study, it's like 55 species around. And then Huia is like the other genus with like kind of like wider geographic range. But then the species account is only like 5. So this is like really bizarre, especially if you compare it to the Bornean population, which is like 13 species. And were to mention that these 13 species were mainly described from the Malaysian part of Borneo. Okay, so then moving forward, this is like kind of like the idea of like the previous phylogeny before my study. And so Amolop is actually like more closely related to the other ranids and not like with the other genera with gastromyzophorous tadpoles. But also like Clinotarsus here is actually not possessing gastromyzophorous tadpoles. But then like the note here is actually like really strange because it's like very short and then like not really strong support. So then like let's move forward to Huia, because then that's the only that's the one that I could have access for, you know. So then like Huia has 5 species, as I mentioned before. So then like what are they? So then the first one was like Huia cafitimpanum, like the type specimen of this genus, which is like described from the Borneo. And then the next time they describe Huia masonai, which is like from Java. But then, so there was kind of like also someone else described Huia javana, but then there are like some contradictory, and then like they decided to like, oh, they look alike, let's just put them together. So it's been like synonymized. And then like the Sumatran species comprises of Huia sumaterana and like Huia modigliani, which actually never been collected since the first original description. And then the 5th species is like Huia melasma, which is like, collected from the Thailand area. But then this one is also like a bit confusing in, not many species that are described. And so how is the Huia position within the ranid gastromyzophorous tadpole? So then here I show you like several studies, like from the beginning. So Stuart was like the first study that kind of like put all the genera together into phylogeny. But then like since then, you see like I put like different set of like markers there, and then the line. But then also like what you can see here is actually like Huia are actually not changing. So Huia is actually like paraphyletics here, according to all of this like studies before my studies. So was it because of the too few samples? That was my like hypothesis, kind of, because there's like in all of this like phylogenies, they only like put like one samples, like one individual in their tree. So I'm really curious, will the addition of more samples would resolve this paraphily? And come to think of this, I also wondering like which one is the true Huia? Is it the cafitimpanum or actually like something else? Okay, so then I decided to just focus on Sumatra just because of the time of my PhD, and also Huia has really wide geographical range. And so why Sumatra? Because Sumatra is actually a topographically heterogeneous, as you can see here. So this is very mountainous region, and therefore they have so many habitats like this, which potential for a ranid with gastromyzophorous tadpoles. But what are they? What can you see there? How many Sumatran cascade frogs? So I mentioned it before, then it was actually only one genus Huia there. But then what are other ranids that often occur in the same type habitat? So there are two. One of them is Odorana, and another one is Chalcorana crassiovis, they call it before. So then among these three, Huia is actually confirmed to have gastromyzophorous tadpoles, and Odorana has no gastromyzophorous tadpole. So what about Chalcorana crassiovis? Well, this one, maybe not, but it's also really problematic. Why? Because they are really abundant. So every time you go to the field, in Sumatra, the stream, you're more likely to find one at least. But then their taxonomic position is really unstable at that time, because it's been moving here and there into different genus, but it's not because they study them, but it's based on implication of the other taxa. So then this is really mainly, I guess, because of lack of samples, they never included in the phylogeny. But then it's also because the type specimen of Chalcorana company, which believed to be also there, are lost. So then they're not sure whether it was they could synonymize that, or the two are exist. So and sort how many species of ranid gastromyzophorous tadpoles are in Sumatra? And yeah, including Huia. Is there any other ranids? And so with this, so where does Chalcorana crassiovis stand in the phylogeny? So to answer this question, I employ the integrative taxonomy approaches, which is combining different sources of evidence. In this case, I use ecology, morphology, and genetic. But I also combine them with large geographical sampling, which means I collect samples from all over Sumatra, but not only for the adults, but also for the larvae. So I did my fieldwork. So this is just a brief idea, how I did my fieldwork, which is literally trying to go across the island and a little bit of Java to collect all a frog looking like a Chalcorana crassiovis and Huia Sumaterana, including collecting the tadpoles. And then I brought everything back in the lab, and I did some genetic and morphological lab, and then run some tree. And then so what was the result? So will my fieldwork, my study really answer these two questions that I mentioned before? Let's see. So to remind you, this is the previous phylogeny of this group of ranids. And then I put this in red because then we wanted to focus on just that. So the Huia Sumaterana is the one in Sumatra, and Huia masona is the one that's supposedly in Java. So my trees kind of not really surprising, but also surprising, because yes, it's actually adding more samples helped. So you, yes, of course. I mean, you see, this is so this is the Sumatra and Java, see? And then so you see more lineages that in that tree compared to the previous one. So then this is basically the result of my study. I mean, don't look at the whole thing, but then just focus on this. So then at least in Java, you already have, two additional species, two species, and then there's three species, or at least occurs in Sumatra. And so this actually answer my second question, which is yes, Huia Sumaterana is not only like the only ranid possessing gastromyzophorous tadpole. So then what about this? And then so this is like actually like the answering my first question about the Chalcorana. So Chalcorana is actually like distantly related, like very far away from the other Chalcorana species. So then this is completely different. So then that's why, so the name you see here is like the new name that I given kind of based on this study. So then let's look at it like a little bit more detail in the next one. So why then Sumatra is a new genus of gastromyzophorous tadpole in Sumatra? Yes, it was like distantly related from the Crassiophysis, so it's definitely not Crassiophysis. So if you zoom out the phylogeny of this, like the tree for this Crassiophysis group from my sample, so then like the tree told like there are like at least like 3 distinct lineages here. And then I also like had sample from the like a topotypic material from like both Chalcorana Crassiophysis and Chalcorana campani. So then both are actually like fell here, which is like, you know, lineages, and there's like not much difference. So then my study kind of like suggests that it could be like synonymized. So then what's next? So this is like the main evidence of why Sumaterana is possessing gastromyzophorous tadpoles, because I got tadpole for the 2 lineages here, like that really like showing this like gastric like sucker here, but then I don't have this one. But then, okay, in terms of this, yes, it's confirmed, but then what else? Like, are these really like real 3 lineages? So then like my morphological study show that like, okay, at least this is, I mean, like this is just like some, including, I didn't really like show the measurement, but then like this is like the main thing that you could possibly tell that they are actually like really 3 different lineages and like really supported the genetic data. So like based on the eyes, and then like here, this is like nuclear pad, and then it's like just like 1, and for this 3rd lineages, it's actually like split here. So then geographically, they're also like distributed into like 3 different lineages, kind of like one is just like restricted to the north of Sumatra, and then like another species, another lineage are like across Sumatra, and then some are just like restricted in the like high elevation. I do not know whether they have here, but I couldn't get any samples, or I didn't go, and that's why I couldn't get any, but it could also be possible that there are occurs there. So with this evidence kind of like supporting to each other, so I have like convinced myself, and then I have enough evidence to like say that like, okay, we need to erect a new genus for like accommodating Crassiophysis group. So, okay, so then this is like the next question, was like about Huia. So then like my study showed that like Huia should be retained for Bornean population only. Why? Let's zoom out again, the tree here. So all of these colors represent the all frogs known as Huia before, and this is like all labels. So then basically at least like 1, 2, 3, 4, 5 study here. But then what's not changing from the previous phylogeny that we know already is that the Huia coffee tympanum. So Huia coffee tympanum is like more closely related to the Bornean population of ranid with gastromyzophorous tadpoles, like a genus, and then not with other Sundaland ranid with belly sucker. And then like a main thing is also, I also have topotypic samples from like Huia Sumaterana, so I can easily ID this as a like Huia Sumaterana clade. But then interestingly, like here I have topotypic samples from the type locality of Huia javana, which was synonymized, you know. But then in this case, it's like match here, and then it's like, so we're pretty confident, and I check also the morphology, it's actually like match the original description. So we actually like resurrect Huia javana, but then in this case, we still do not know where is Huia masoni. So that's the only problem. And then I try to check it with this lineages, but then it's not Huia masoni. Why? Because Huia masoni doesn't have like inner metatarsal tubercle, no, outer metatarsal tubercle. And this one has, so then it must not be Huia masoni, but where, I do not know, because I also don't have the samples. And then like the type specimen was like ambiguous, kind of. So then that will lead me to have this postdoc, where I can explain later. But then let's focus on this. So with this is just like p distance of like all of this clade that I mentioned. So then basically this clade here I mentioned here is actually like more as a, like Huia melasma, and another Huia is actually here, which is like similar to like genus to genus distance. So then in this case, I suggest that it would be like a very different between these lineages. And then another evidence would be like, again, from tadpoles, because tadpole is like so powerful. And I measure like a much more tadpole from like a Singapore museum. And I think I have like more than 100 tadpoles examined like in that during the 2 weeks. So then basically this is like the result, and then they're like a clear difference. And then especially for this Sumatra and Javan clade here, they have like the same shape of jaw's head, which is like M shape here. And then like the Bornean one is just like V shape. And in Laos and Thailand, no idea, no one studied that, and then no one really know. And then like, yeah, so everything is like super clear that they are like completely different. And then so it's matched with the genetic. And that's why we decided to, okay, let's just like retain like a Huia just for the Bornean population, especially because the type species was like from here, like from Borneo. And then, so what shall we do with the rest? So for the moment, like this one is like pretty confident, like Sumatra and Java is Sumaterana, but then the Laos and Thailand, it's like here, I mean, like we still do not know, and then there's like not clear evidence. So then for the moment, we decided to just like tentatively put them in a Wijayarana just based on like this tree here. Okay, so then this is just like a wrap up for like what I questioned at the beginning. Like, okay, so then basically my study, like adding more samples, is actually like can tell that like Chalcorana crassiophis is a member of gastromyzophorous tadpoles, and we have at least 6 species in Java and Sumatra, only. And then the true Huia is actually like Huia capitimpanum. And then like highlight of this study is that like integrative taxonomy approaches should be combined with large geographical samples, but not only for adult, but also tadpoles. Okay, so then I kind of like already have or resolving the phylogeny of this like group of frogs, then I try to answer some biogeographical question that I'm interested in, because I already also like a set of data, which is like very interesting. And then come to think of it, because they're like very dependent on like a cascading habitat, then I'm really interested to see like the correlation of this like the precipitation pattern of this like with this like river system that was like occurred in Sumatra during Pleistocene. So then you see there are like 4 river system in Sundaland back then. And I want to see like whether the diversification pattern of the Sumatran ranid with gastromyzophorous tadpoles was influenced by the Paleo River. And this question was intrigued, like, so there was previous study, but then like on fish. So like within these two fish, and then they said, oh, actually like this river system is aligned with the distribution pattern of like these two genera of fish. So then this is like the tree from my data set. So then I mapped the distribution pattern into a phylogeny. You see like this color-coded maps and also like tree here. It's like basically like showing this is like where I was collected. And then like this is like represent, like the river system, which like 1, 2, 3, 4, 5. But then forest was not really defining the river system that are like flowing into the Indian Ocean. And then so I wasn't really sure whether like all of the river system that he defined was also like flowing in this side. So then I decided to put like additional category here. So it's actually there's like no correlation at all, or like no, I couldn't really see strong correlation here that their pattern of distribution is influenced by this river system. Why? It could be because this is like the river system was too young, and then like the frogs already colonized Sumatra way before that. But then I also come up with like some hypothesis that why it works in fish, because fish live like entirely in the river, like in the water, while frogs has biphasic life cycles. It's like during the adult stage, they are like terrestrial, so they could like move around further, and then that could probably like change their distribution pattern. However, it's very interesting that I actually like found some pattern on their like distribution. So I tried to map them, then like see how they actually like, whether they have like some genetic structure on that. So then that actually like, there are like at least, I mean, like, I'm still not sure whether this is just like really like two like north and south lineages only, or they're like more than that, because actually like there are like some overlapping here in the middle that wasn't clear whether like that is like the barrier, like the border of between the two, or they're like its own. But this this partitioning has also already like studied in other taxa, and it's like more or less it like have similar pattern. For example, and like tree frogs, like monkeys and like mountain dragons, so they actually have like the north-south, but then like some of them are decided to, like this, they have more categories, and like they try to come up with like a scenario how to explain that, and then like probably like they're like, like paleoclimatic is like the most possible scenario for that. But then like for monkey, they actually like think that Toba supervolcanic eruption was like the main thing that segregate the genetic of like this monkey. But then like for this recent paper, they also like think that like, oh, there might be like more complex than that. Maybe like some also like needs partitioning and everything. So then I haven't go that far for my frogs, so I'm not sure, but it also be like more complex. So I tried to come up and like when was like these two genera reach Sumatra. So they actually like arrive in Sumatra, like my time tree like showed it's like around Oligocene, like within 25, 27 million years ago. And that is actually like matched with like the picture back then, like the like more land masses, like connecting between like main Asia and also like Sumatra and Java. But then interestingly, my tree showed that there was like a return phase of like a Huia at least. So Huia like went to Java, like so from the yeah, mainland to Sumatra and then like all the way to Java in the northern tips. But then at some point, they also like some of them like make it back to Sumatra during, yeah, like around like 1642 million years ago. So I run a little bit to Biogeography and then like to explain how that was like actually like happening, why then like we can have like such distribution. So then this is just like a hypothetical scenario that I got like from Biogeography, that like a series of divergent vicariance and even just like a copy range, like distribution range copy. So yeah, that is like a little bit of biogeography, but then like this is also like one of why then I'm here like doing my postdoc, because like so many more questions to answer. So this is like the last phylogeny that I have, and like what was like the next question that I would like to answer. So of course the Huia melasma, the mainland population need to be confirmed whether they have gastromyzophorous tadpoles or not, and then like how much diverse they are. And also for Huia caffeotympanum, are they really a monotypic genus? Because at the moment it's just like one a species under this genus. And most importantly, this one, like the Sumatran clade, we need to like confirm like what is the identity of this tree, and then of course also like to answer these two species whether they are actually like valid species or not, because I mentioned it before that like Huia amazonite is not confirmed yet where they're in the phylogeny, and then the only way to do it is like for the two, or just like to obtain their DNA from the type materials and then put them like in the phylogeny. And that's why I doing my postdoc here, I joined a collaboration between MPZ and IYB, and then funded by the European Commission to really obtain using high throughput sequencing to obtain a DNA sequence for like resolving the phylogenetic problem of this group. So that was a brief of what I have done in this group, and then like many more things that I wanted to know, but then those studies has won't be possible without like support for like so many like fundings and especially people. But then in this case, I would use this opportunity to really thank Jim because yeah, he mentioned that like I was introduced into the field herpetology because I was joining like his expedition, and then that brought me to where I am here. So then like he's like one of the most important people in my career in herpetology. So thank you so much, Jim. And that would also like lead me to more acknowledging myself, like who I am and then where I am in my career journey, because it's been a long journey for me and it wasn't easy coming from Indonesia, a small village, and then I got to go to like move to another cities for school, and then made it to move to Germany and then to the US. Well, I do not know where next, but we will see. But yeah, with this, I actually like really glad that I share this like experience with another peer, like best friend, colleague that are also like in the same level of career. And then we actually like decided to join forces to create this project, like we call it like the Global Women in Herpetology project, because we want to connect like all women herpetologists across countries and cultures. So we have the two main programs. One of them is like the book. So the book will be like just a story compilation of 50 stories from like 50 women from 50 countries. So one woman per country, which is like 25% of world countries. Yes. And yeah, and then we wanted to make it like a more contribution, and then not just like sharing our stories, but we wanted to also like support like a young student, especially woman, to pursue their career in herpetology, so that like all book sales will be for travel grant for like all female student to attend a herpetological conference. And then the second program is like a network directory. It's like a database basically for all women herpetologists in the world. And then so you can also find them like either seeking collaborators or networking and so on. And yeah, so on behalf of all female herpetologists in the world, please feel free to learn more about like our project and donate so we can publish the book, and then make it real that we want to support like more students that are probably like me. And yeah, please enjoy the life in the fast flow. And I'm really thank you for your attention.
Plenty of time for questions. I'll let you call on folks yourself.
Okay. Yes, Becca?
Yeah, so you get material from this egg specimen, and how's that going? So you will sequence it?
Yes, so it's like ongoing. I just doing the library and the library is almost like done. Yeah, so maybe I can come back and tell you more about this in the next launch seminar.
So I was confused on how many times the sucker evolved in the tadpole. Is that just once, or did it evolve multiple times? Was it lost in some groups?
Yes, that is really good question. I did not explain that, but glad you asked. So we can come back to the, where was the tree? Yeah, we can use this, whatever. Okay, so then this is amolops, right? So this is like more closely related to this. So that could possibly like start in here, and then like it's like somehow like lost, but then you have it again here, but then it's also like probably lost again because you have Clinotarsus here. So it's probably like multiple time evolution.
I guess my question was, do you think it evolved just once and then was lost in various lineages? Or can you distinguish between that and the possibility that it evolved multiple times?
That was like the hypothesis until now, but then that's why I really wanted to, like have this like project, like my postdoc project. So then like with that, like a stable phylogeny, then I could like run, analysis how many times, or like how do they evolve actually? Because the problem was here, I mentioned it before. And then if you can see it in this other tree also, like in the previous trees, yeah. So then that actually like it's always, like moving. Let's just say like Clinotarsus. So in this tree, Clinotarsus is closely related to Huia melasma, but then with like very low support value. And then like Clinotarsus here is actually like closely related to like a Sumatran Javan species, a slightly higher value, but then like still not quite. And then like here it's like kind of like similar with that, but then, oh, they don't have it here. But then like even in my phylogeny, it's like never really like strong support everywhere, and it's always like placing here and there. But then I'm not sure, because then I mean like all of this studies like has more markers, and then like ranging from like shorter and shorter length of sequence, and then like longer sequence, and then like, but then like their problem was like too little samples. But then I tried to add my samples, like more samples, so then I probably like have more comprehensive data for this data set. I also include like all genera that possess gastromyzophorous tadpoles. but then it still doesn't change. So then that is like possibly like, I don't know, very like faster for genes.
I wonder if the ecology of the frogs or the tadpoles can tell you something about the likelihood of convergent evolution versus loss. Like, I don't know Clinotarsus, but does Clinotarsus, do they occur in fast flowing streams where
Yes.
a sucker tadpole would be useful, but they don't have one? And that might suggest that multiple origins is more likely. Or, but if Clinotarsus is in ponds or in slow moving streams, then loss might seem more likely for them. So I'm wondering if you can use that information to help.
Yeah, that is interesting view. But then I found Clinotarsus when I visited India, and then they're also in a like kind of like fast stream. Yeah,
Did you see their tadpoles?
I saw. Yeah, without
Or sucker?
gastro. Yeah.
Do you
But
see
the-
sucker tadpoles in any other frog groups? Or is it just?
So
Yeah.
just in this like Asian ranids that I'm studying, and some neotropical bufonids.
But then
Like
there are
ask-
oral suckers in some tadpoles as well.
Yeah, oral suckers are normal. This is not normal. Yeah, and then
Stomach suckers.
yeah, and then yeah, they do also. I mean, because even in Indonesia, even in Sumatra, they also have buffonid, and then buffonid has like oral sucker. And then I also saw some buffonids in this like fast flowing, river where I found them. But then they just like have different, like way of adaptation. And then I guess that's also probably like niche partitioning, because the tadpoles probably just stay in the like bank, while this one, most of them I found it like in the river, like literally like under the swift. Although like, so it's like literally like have to be like with the swift, and it's like under. You saw the video before. So then that's why that like sucker mount is like, well, yeah, this is like not too extreme. My mouth can handle this. So well, that's why not, yes.
Can I ask another question? Just
Yes.
It occurred to me when you were describing this.
Okay.
Do you think that having the sucker allows them to occur in parts of the stream that are where they're less susceptible to predators? Do you think that's one of the advantages of this, or is it just that that's where they need to be in order to get the food? Just looking at it in the stream like that, it seems like it'd be hard for a fish to negotiate that potentially. Although maybe Chris Martin would reject it.
Maybe he can help me answer the question. But yeah, but here's, okay, no, yeah, but here's my answer. That's probably like one of the reason. But then I can give you a little hint what I saw in the field. So then that's probably true. That's like one way of like he's like avoiding like predator. But then Wijayarana Wijayarana tadpoles is actually like not always like under the swift. I mean, yeah, but then so the adults are a little bit more tolerant with like how strong the current are. So although it's like not that strong, you can sometimes find them. But for Sumaterana, it must be in the river, and then I also need to have boulders, not just like small rocks. It doesn't work for like a Sumaterana. I saw them like less abundant than in the stream with like boulders. And also interestingly, I found like the two tadpoles from the two genera occupying the same rocks. And then so my future question was also like to know like whether they're really eating the same algae or not. If so, then they must be competing to each other. And then additionally, I also saw observe fish, because some fish has also like sucker, and then I also saw them occupying the same rock. So that's probably more complex, not only like competing to the other genera, but also the other taxa. Yes.
Yeah, it's really cool adaptation. I mean, it reminds me, have you heard of the waterfall climbing gobies of Hawaii?
Yeah.
Yeah, that's
Yeah,
only-
one of them is goby. I saw them.
Yeah, I mean, there's modified pelvic fin suction cups all over fishes. the only sort of motion I know of, like your tadpoles, they have the same sort of oral suction and then pelvic plate, like an inchworm. It's really cool to sort of compare locomotion
Yeah.
between the two.
Yeah,
Kind of a cool example.
yeah. I thought about that already.
So yeah, fishing.
Fishing everything, right?
Yeah.
All right, well, let's thank Umi for a great talk.
My pleasure. Can come to me to talk more.
Great job.
Yeah, you got to